Long before bedrooms, before shutters and curtains, before the hum of electricity, humans lay down beneath the stars. Their sleep was not sealed away in walls, but bound to the vast expanse of the night sky. The earliest humans drifted into unconsciousness with constellations overhead, their minds tuned to patterns of light, and darkness so ancient that even today, under layers of architecture and technology, the brain has not forgotten them. Modern psychology and neuroscience increasingly show that this connection to the stars is not a romantic artifact of history, but a structural element of human biology. To sleep without the sky above is, in a sense, to break a continuity millions of years old. When the sun sank beyond the horizon, the world did not dissolve into absolute blackness. Moonlight and starlight traced delicate shadows across landscapes, giving the night a rhythm distinct from day, but equally consistent. This interplay between darkness and faint illumination shaped the earliest circadian cycles, the 24-hour oscillations that still govern every organ system. Long before anyone had words for melatonin, serotonin, or the suprachiasmatic nucleus, the human organism calibrated itself to the waxing and waning of celestial light. Even in the absence of a mechanical clock, the night sky was an unbroken metronome, and the mind came to rely upon it. Neuropsychological studies suggest that environmental cues, what researchers call Zeitgebers, are crucial to synchronizing the circadian system. Today, most people think of sunlight as the dominant Zeitgeber, yet for much of evolutionary history, the stars and moon provided the secondary framework that distinguished one phase of night from another. Without this subtle contrast, darkness becomes formless, and for the brain, formlessness means uncertainty. Anxiety increases, vigilance rises, and sleep is delayed. The earliest hominids, exposed to predators and environmental hazards, could not afford such vulnerability. Their rest depended on an equilibrium between safety and surrender, an equilibrium etched into neural architecture by countless generations who fell asleep with the cosmos as their ceiling. What is striking is that the psychological effects of starlight remain detectable even in modern contexts. Experiments with controlled lighting environments reveal that individuals exposed to low-intensity natural night light, levels equivalent to starry skies, report not only faster sleep onset, but also a heightened sense of calm before sleep. The phenomenon resembles what clinical psychology calls conditioned safety signals, cues that indicate an absence of threat. Just as a child clings to a ritual object or a soldier relaxes at the sound of a ceasefire whistle, humans learned to associate the glittering canopy of stars with an environment momentarily free of immediate danger. In the absence of that canopy, unease often lingers without conscious explanation. This hidden dependence on the stars is not confined to evolutionary theory or controlled studies. It has seeped into every layer of human culture. From the Epic of Gilgamesh to the dream songs of the Navajo, myths describe slumber beneath the stars not simply as rest, but as passage into another state of being. The very idea of falling asleep was often expressed through metaphors of descent into the sky, as if dreams themselves were drawn down from constellations. The human brain, predisposed to search for patterns, used the regularity of the night sky to anchor not only its physiology, but its imagination. To lie down beneath the stars was to be reassured both biologically and symbolically, to experience a rare alignment between neural chemistry and cultural meaning. Modern bedrooms, by contrast, are sealed environments of controlled temperature and artificial darkness. They mute the very cues that once prepared the nervous system for rest. The result is a paradox. In trying to eliminate external disturbances, we may have removed the signals our ancestors relied upon to let go of wakefulness. Sleep psychologists note that many contemporary sleep disorders, from insomnia to delayed sleep phase syndrome, reflect not an excess of stimuli, but a deprivation of natural temporal anchors. Behind blackout curtains, the brain remains suspended, uncertain of when to yield, caught between its prehistoric wiring and its manufactured environment. This is why a simple act, stepping outside at night, looking up before bed, can produce disproportionate effects. A brief exposure to the sky, even in polluted cities where only a handful of stars are visible, provides the nervous system with a context it has been conditioned to recognize for millennia. In that fleeting moment, the body receives permission to release its vigilance. Sleep researchers such as Kenneth Wright at the University of Colorado have demonstrated that even short camping trips, where participants fall asleep beneath open skies, rapidly recalibrate circadian timing and restore deeper sleep stages. Such findings hint at a truth that unsettles the modern narrative of progress. No matter how advanced our environments become, the body remains anchored to rhythms written in the night sky. To understand why humans cannot truly sleep without the stars above, one must begin not with contemporary disorders or modern solutions, but with the vast span of evolutionary time in which night meant exposure to the cosmos. The nervous system that craves security, the glands that secrete hormones at twilight, the networks of neurons attuned to faint variations of light. All of these are legacies of those countless generations for whom sleep and stars were inseparable. The structures remain intact, waiting for a signal that no roof or electric bulb can replicate. And so, even today, when the room grows dark and the ceiling replaces the heavens, the brain quietly remembers what it has lost. The absence is not always noticed, but it is always registered. Sleeplessness becomes, in part, the echo of an evolutionary dissonance, a body searching for patterns it once trusted but can no longer find. For the greater portion of human existence, there were no roofs to separate consciousness from the open sky. Archaeological evidence from early hominid sites suggests that shelters were temporary, porous structures, providing modest cover against wind or rain, but leaving the night overhead unobstructed. In such environments, sleep was not an isolated act within an enclosed chamber, but a phase of life embedded within the landscape. The darkness above was constant, yet never absolute, filled with the shifting positions of stars and the slow wheel of the Milky Way. This canopy of movement provided a backdrop against which human biology adapted, and its imprint can still be traced in the mechanisms that regulate rest. The paleontological record places Homo erectus in Africa nearly 2 million years ago, and while much attention has been given to their tools and diet, their sleeping environments were no less critical to survival. Fossilized bedding at sites such as Border Cave in South Africa, composed of grass layers dating back nearly 200,000 years, indicates deliberate preparation of sleeping areas, but always in open or semi-open spaces. Fire provided localized security, but the sky remained unshielded. Night after night, those early humans closed their eyes beneath the stars, and the nervous system calibrated itself to rhythms reinforced by celestial regularity. Ethological comparisons with other primates are instructive here. Great apes construct arboreal nests, often leaving them exposed to starlight and moonlight, their cycles of rest shaped by nocturnal cues. Humans diverged, moving more frequently to the ground, but carried the same neural predispositions. The reliance on external celestial markers for timing did not diminish. It deepened. The visual cortex, among the most developed regions of the human brain, was persistently engaged by star patterns, and over generations, this engagement imprinted itself as part of nightly orientation. To sleep under a sky alive with constellations was not merely a passive condition, but an active part of sensory regulation, one that provided assurance of continuity and time. Evolutionary psychology suggests that threat detection and rest exist in a precarious balance. Sleep requires surrender of vigilance, but such surrender is only possible when the environment offers consistent cues of stability. The stars, fixed in position relative to one another, became symbols of permanence in a world of shifting dangers. This stability was not an abstraction. It shaped survival strategies. Groups who learned to time their activities by the movement of celestial bodies were better prepared for nocturnal predators, migrations, and resource gathering. The capacity to trust the sky became intertwined with the ability to achieve restorative sleep. Those unable to find calm in the patterns above would have struggled with fatigue, impaired cognition, and reduced chances of survival. The archaeological record of Paleolithic cave art adds another dimension to this relationship. Many of the oldest paintings, dating back over 30,000 years, depict star clusters and lunar phases alongside animals and human figures. The integration of night sky imagery into early symbolic systems suggests that celestial observation was not restricted to navigation or ritual. It was lived experience. Sleeping beneath these same skies likely reinforced their psychological weight, turning nightly exposure into a foundational rhythm of both biology and culture. Anthropological studies of contemporary hunter-gatherer societies provide a window into what earlier human sleep might have resembled. Groups such as the Hadza of Tanzania or the San of the Kalahari Desert still sleep largely outdoors, with minimal artificial barriers between body and environment. Ethnographic accounts note that individuals drift off while gazing at the stars, often using constellations to track seasonal changes and nightly progression of time. Sleep in such contexts is segmented but not fragmented. People wake briefly, tend to fires or children, and then return to rest, always under the same vault of light. The night sky remains a participant in the cycle, its presence shaping both timing and psychological reassurance. This evolutionary backdrop clarifies why modern humans, even in urban settings, often report a peculiar restlessness when entirely cut off from natural sky exposure. The brain retains mechanisms that expect certain environmental cues before yielding to unconsciousness. Laboratory studies simulating prehistoric lighting environments show that when participants are exposed to a gradual dimming that mirrors sunset, followed by the faint luminance of starlight, their sleep onset occurs more quickly, and their sleep stages exhibit greater stability. Such findings align with the hypothesis that the human nervous system evolved to depend on celestial transitions as part of its nightly calibration. Yet the evolutionary pressures went beyond simple light exposure. The stars also provided orientation. Before the invention of maps or compasses, navigation by constellations was a matter of survival. Knowing how to return to camp, when to begin a migration, or how to avoid disorientation in vast landscapes. Sleep under the stars meant constant reinforcement of spatial awareness. Even in unconsciousness, the last sensory input before eyelids closed was often the arrangement of constellations. Neurocognitive research suggests that the hippocampus, critical for spatial memory, may have drawn from these nightly exposures, strengthening its role in mapping both land and sky. Over time, these associations hardened into neural expectations. Just as the sound of waves conditions coastal dwellers to rest, or the rhythm of crickets lulls inhabitants of temperate zones, the presence of starlight became a nearly universal cue for relaxation. The absence of such a cue does not erase the expectation. It leaves it unsatisfied, creating a void that the nervous system struggles to reconcile. Modern individuals who sleep in entirely sealed environments may not consciously perceive the difference, but their bodies respond with lingering hyperarousal, a residue of vigilance that never entirely subsides. This evolutionary inheritance raises a quiet paradox. Humanity's most advanced constructions have systematically severed the nightly contact with the very sky under which its biology was shaped. Bedrooms may provide security from external threats, But they also deprive the body of signals it has been conditioned to expect for hundreds of thousands of years. What began as survival in the open has become dissonance in confinement. The nervous system still seeks orientation in the patterns above, even when those patterns are hidden from view. As modern life accelerates, this tension grows more pronounced. Artificial ceilings, unbroken darkness, and digital glow displace the ancestral canopy, yet the brain continues to search for its lost framework. The result is not only difficulty in falling asleep, but a persistent sense of incompleteness, as though some element of the environment remains missing. The mind, after all, does not evolve at the pace of architecture or electricity. It carries the traces of its formation across millennia. Each night spent without stars leaves an invisible residue, a reminder of an alignment once taken for granted, but no longer assured.
Deep within the brain, a cluster of approximately 20,000 neurons operates as the master regulator of time. Known as the suprachiasmatic nucleus, or SCN, this structure sits above the optic chiasm, where signals from the eyes intersect before traveling deeper into the brain. Its location is not accidental. The SCN depends on light exposure to calibrate its rhythm, orchestrating a daily cycle that governs everything from hormone secretion to body temperature. It is this rhythm, the circadian clock, that makes sleep possible in alignment with environmental patterns. For early humans, the night sky provided more than a backdrop. It was an integral part of this regulation. The SCN receives input from specialized retinal ganglion cells containing melanopsin, a photopigment particularly sensitive to short-wavelength blue light. Sunlight saturates these receptors, but after sunset, when only the muted glow of stars and moon remains, the signal shifts. The absence of strong blue light, combined with a faint scattering of natural luminance, tells the SCN to trigger downstream processes. Melatonin secretion begins, cortisol levels decrease, and the body enters a phase of readiness for sleep. This nightly cascade did not emerge spontaneously. It was sculpted over evolutionary time through exposure to consistent celestial cycles. Chronobiology has revealed that these mechanisms are highly precise. Experiments in temporal isolation chambers, such as those conducted in the mid-20th century by Jürgen Askhoff and his colleagues, demonstrated that humans kept in complete darkness or under constant dim light exhibit free-running circadian rhythms slightly longer than 24 hours. Without external cues, the clock drifts. What realigns it to the environment are natural cycles of light and dark. Before artificial light, the stars and moon acted as stabilizing signals that prevented this drift from pulling physiology out of sync with the planet's rotation. The influence of starlight on circadian rhythms might seem negligible compared to the power of the sun, yet neurophysiological evidence shows that even extremely low levels of illumination can reset the SCN. In fact, the threshold for circadian entrainment lies within the range of natural nocturnal light. Measurements of night sky brightness in unpolluted regions fall between 0.001 and 0.1 lux, a range sufficient to produce subtle but measurable effects on circadian timing. Modern light meters confirm what the nervous system already knew. The night sky is not dark emptiness, but a calibrated field of information, and the brain is sensitive to it. What makes the SCN remarkable is its influence over the entire organism. Once activated by shifts in light, it coordinates peripheral clocks located in tissues throughout the body. The liver, pancreas, heart, and even adipose tissue contain molecular clocks that anticipate daily fluctuations in activity and rest. Gene expression cycles, driven by transcriptional translational feedback loops involving clock, BMAL1, PER, and Cry proteins, rise and fall in synchrony with SCN signals. When celestial transitions guided these processes, the system remained coherent. The timing of digestion, metabolism, and immune activity aligned with patterns of exposure to sky and darkness. Without that alignment, desynchrony emerges, leaving physiology vulnerable to fragmentation. Neuroscientific imaging has revealed how closely tied this process is to mood and cognition. The SCN communicates with the pineal gland to regulate melatonin, but it also projects to the amygdala, hippocampus, and prefrontal cortex. These connections influence emotional regulation, memory consolidation, and executive function, all processes that depend on adequate sleep. When circadian rhythms misalign, individuals report heightened anxiety, impaired learning, and depressive symptoms. Psychiatric studies link circadian disruption to bipolar disorder, seasonal affective disorder, and major depression. The absence of natural sky cues, once dependable anchors, becomes more than a physiological inconvenience. It destabilizes the very foundations of mental equilibrium. Historical evidence underscores how attuned societies were to these rhythms. Ancient agricultural communities developed calendars based not only on solar cycles, but on stellar risings and settings. The heliacal rising of Sirius, for example, signaled the flooding of the Nile in ancient Egypt, determining planting schedules and rituals. These observations were not merely practical. They reinforced the body's alignment with environmental cycles. By embedding sky patterns into cultural practice, societies ensured that individuals remained synchronized with celestial rhythms, even before the science of circadian biology existed. Yet the SCN does not function in isolation. It is exquisitely responsive to competing signals. Artificial light sources, introduced only in the past 2 centuries, deliver wavelengths far more intense and prolonged than starlight. Exposure to these sources at night suppresses melatonin and delays circadian phase, producing what chronobiologists call social jet lag, a chronic misalignment between biological rhythms and environmental demands. While the body evolved to track faint shifts in the night sky, it now confronts abrupt exposures to incandescent bulbs, LED screens, and neon signs. The nervous system struggles to interpret these cues, oscillating between evolutionary expectations and technological intrusions. The consequence is a widening gap between internal time and external environment. Studies measuring dim light melatonin onset, a marker of circadian phase, show that individuals living in brightly lit cities often experience delays of several hours compared to those in rural, starlit settings. This delay translates into difficulty initiating sleep, shortened rest periods, and altered REM cycles. What the SCN once regulated effortlessly, using cues from stars and moon, is now in constant negotiation with competing stimuli. The body's central pacemaker continues to function, but its harmony with the cosmos has been disrupted, leaving its signals muted, uncertain, and easily overridden. As research in chronotherapy expands, scientists have begun to explore ways of realigning circadian rhythms through controlled exposure to natural cycles. Experiments involving wilderness immersion, where participants abandon artificial light and sleep under open skies, demonstrate rapid recalibration of circadian phase. After only a few nights, melatonin onset shifts earlier, cortisol rhythms stabilize, and subjective sleep quality improves. These findings suggest that the circadian system retains its responsiveness to ancient cues, even when submerged beneath modern distractions. The architecture of the SCN has not forgotten its relationship with the sky. It waits for signals that the modern environment rarely provides. Yet the question remains unresolved. If the SCN can adapt to new light regimes, why does the absence of stars continue to exert such subtle pressure on sleep and mood? Why does exposure to the open night sky produce a depth of rest that artificial environments cannot replicate? The answer lies not only in the mechanics of circadian entrainment, but in the broader network of neurochemical and psychological processes that converge in darkness. The circadian clock initiates the transition, but the quality of that transition depends on forces that extend beyond the SCN itself, pulling the body toward a state of surrender that remains elusive when the night above is concealed. When the suprachiasmatic nucleus interprets the fading of light, it signals the pineal gland to begin releasing melatonin. This small, pine-cone-shaped structure, situated deep within the brain, has long been associated with mystery. René Descartes once called it the seat of the soul, a symbolic reflection of its centrality in regulating states of consciousness. Today, neuroscience describes it as a key regulator of circadian timing, yet the influence of melatonin extends well beyond simple sleep induction. Its secretion represents the body's recognition that night has truly arrived, and for most of human history, this recognition depended on the purity of natural darkness punctuated only by starlight. The biochemical cascade is precise. As evening approaches and the intensity of light falls below the threshold detected by retinal ganglion cells, sympathetic fibers from the superior cervical ganglion release norepinephrine onto pinealocytes. This stimulates the enzyme serotonin N-acetyltransferase, which converts serotonin into N-acetylserotonin, later methylated into melatonin. The rhythm is tightly bound to environmental light conditions, but for countless generations, those conditions were shaped by the slow dimming of the sky and the faint persistence of celestial illumination. The predictable rise in melatonin coincided with the appearance of stars, a connection the brain came to expect night after night. Melatonin's role extends into multiple physiological domains. It lowers core body temperature, reduces alertness, and promotes synchronization between peripheral clocks in organs and the central rhythm of the SCN. The hormone also functions as a powerful antioxidant, scavenging free radicals and protecting neural tissue from oxidative stress. Clinical studies show its involvement in immune regulation, with elevated nocturnal melatonin linked to increased production of natural killer cells and enhanced adaptive immune responses. In evolutionary terms, this meant that rest under starlit skies was not only restorative in a psychological sense, but also essential for physical resilience against disease and injury. The intensity of starlight may appear too weak to matter, yet measurements suggest otherwise. Lux levels beneath a natural night sky, particularly with a full moon, are sufficient to modulate melatonin production without suppressing it completely. This faint illumination provided a balance. Enough visibility to maintain orientation and safety, but dim enough to sustain hormonal release. Modern studies demonstrate that even exposure to light as weak as 5 lux can delay or suppress melatonin secretion, a brightness exceeded by most artificial environments. By comparison, the light of stars typically falls below 0.01 lux, a range within which melatonin rises unimpeded. The alignment between neurochemistry and celestial conditions was finely tuned, and the nervous system adapted accordingly. Historical accounts reinforce how closely human rest was bound to these rhythms. In pre-industrial Europe, segmented sleep was common. Individuals fell asleep shortly after dusk, awoke during a watch in the middle of the night, and then returned to rest until dawn. During this interval of wakefulness, often lasting an hour or more, people prayed, wrote, or conversed quietly under the muted glow filtering in from outside. Diaries and medical texts from the 17th century describe this first sleep and second sleep as ordinary, regulated by natural darkness rather than disrupted by insomnia. The gradual ebb and flow of melatonin across the night supported this rhythm, shaped by a sky that signaled both rest and pause without extinguishing its presence. Cross-cultural evidence reveals a similar pattern. In agrarian societies, sleep aligned with lunar phases, adjusting to brighter nights when work could extend longer into evening. The modulation of melatonin under moonlight produced subtle changes in alertness, enough to allow extended social or ritual activity without fully disrupting circadian order. Recent chronobiological research supports this. Studies conducted in rural Argentina show that melatonin onset occurs later during nights leading up to a full moon, a reminder that human biology remains sensitive to nocturnal celestial cues even when daily life is dominated by artificial light. The implications for modern sleep disturbances are significant. Insomnia, delayed sleep phase disorder, and other circadian-related issues frequently correlate with insufficient melatonin signaling. Artificial light, especially from LED screens emitting blue wavelengths, suppresses melatonin more aggressively than starlight ever could. Laboratory research at Harvard Medical School has shown that exposure to blue-enriched light before bed can reduce melatonin levels by up to 80%, delaying sleep onset by hours. What once occurred seamlessly under the open sky has become a delicate process vulnerable to disruption by a single misplaced light source. Supplemental melatonin has become a widespread intervention, yet it addresses only part of the problem. Pills provide exogenous hormone, but they do not replicate the psychological and sensory environment that naturally accompanied its secretion for millennia. The alignment of body and cosmos cannot be recreated by pharmacology alone. The stars offered more than an absence of light. They provided a patterned canopy of continuity. The faint glimmer overhead signaled safety and permanence, cues that reinforced the body's willingness to surrender to sleep. Without those signals, melatonin may rise, but the deeper sense of alignment remains incomplete, leaving rest less restorative. The pineal gland itself retains echoes of this ancient bond. Unlike most brain structures, it is not shielded by the blood-brain barrier, allowing it to respond directly to circulating molecules in the bloodstream. Its calcification in modern populations, often accelerated by environmental factors such as fluoride exposure or disrupted circadian cycles, raises questions about the cumulative effects of disconnection from natural night. The decline of pineal function in such contexts mirrors the decline of contact with starlight, as if biology itself reflects the absence of its ancestral partner. When individuals today seek deeper rest by escaping urban light pollution, camping in remote deserts, lying beneath unbroken skies, the improvement is often immediate. Sleep becomes heavier, dreams more vivid, and waking more refreshed. Objective measurements confirm these subjective impressions. Polysomnography under natural night conditions shows longer durations of slow-wave sleep and more consolidated REM cycles. These outcomes are not easily explained by melatonin levels alone. They suggest that melatonin acts as the initiator, but the surrounding sensory environment determines the depth and integrity of the state that follows. The alignment between chemistry and sky was never perfect. It fluctuated with weather, seasons, and geography. Yet within those fluctuations, humans developed resilience. The faint certainty of stars provided just enough continuity for the body to yield to unconsciousness. What remains uncertain is how far modern life can drift from these conditions before the accumulated strain reshapes not only the architecture of sleep, but the stability of waking consciousness itself. To sleep beneath the night sky was never a passive act of surrender. It was also an act of orientation. The human brain is wired for spatial awareness, with the hippocampus forming intricate maps of both immediate surroundings and distant landmarks. Before compasses, before written charts, the heavens served as the most reliable points of reference, a framework that anchored individuals within an otherwise shifting environment. The very last images absorbed before sleep were often constellations, their stable arrangements offering reassurance not only for navigation but for survival itself. Navigation by stars has been documented across continents and epochs. Polynesian voyagers steered their canoes across vast oceans using constellations as guides, memorizing star paths that rose and set along the horizon. Ancient Arabian caravans followed the North Star to traverse deserts where landmarks shifted with the winds. The Greeks formalized these patterns in celestial charts, embedding orientation into myth. In all cases, the stars functioned as fixed markers, reducing uncertainty in landscapes otherwise defined by impermanence. The link between sleep and orientation lies here. When the mind settled beneath a predictable canopy, it knew where it was, and where it could return when waking resumed. The hippocampus, critical for spatial memory, is also intimately connected to sleep. During slow-wave stages, it replays sequences of neural firing associated with navigation and exploration, consolidating them into long-term memory. Exposure to constellations before rest may have reinforced this process, aligning neural activity with recognizable celestial anchors. Studies on rats navigating mazes show that hippocampal place cells fire in sequences during sleep, effectively rehearsing the path taken. For humans, the stars may have offered the broader frame in which such rehearsals took place. Falling asleep while oriented to the heavens was not simply comforting. It primed the brain's navigational system for the demands of waking life. Fear and vigilance also played decisive roles. Darkness without orientation fosters anxiety, heightening amygdala activity and delaying rest. The presence of the stars counteracted this uncertainty. Knowing that constellations remained in fixed positions and that their movements across the sky were predictable, created an external stability that the nervous system could trust. Ethnographic accounts of pastoral nomads describe this reliance vividly. Herders in Central Asia speak of sleep coming only when they can see the Great Bear above them, a celestial guarantee of their camp's position within a vast steppe. The stars functioned as an external nervous system, absorbing the burden of constant vigilance and permitting the body to relax into sleep. Even the illusion of orientation carries profound effects. Psychologists studying wayfinding have demonstrated that individuals deprived of external cues, such as in dark rooms or fog, quickly lose their sense of direction, often walking in circles without realizing it. This disorientation triggers stress responses, elevating cortisol and increasing heart rate. In contrast, a single fixed point, a star, a fire, a visible landmark, restores calm. The nervous system evolved in an environment where the stars provided this function nightly, making them not just navigational aids but psychological stabilizers. Sleep was more accessible when orientation was secured, and in the absence of that security, rest remained shallow or fragmented. Religious and mythological traditions reflect this deep association. In many cultures, stars were not simply lights but guardians, fixed presences that watched over sleepers. The Navajo described the night sky as a protective blanket, its patterns woven by divine hands. Ancient Egyptians aligned their pyramids with celestial poles, embedding orientation into monumental architecture. Such beliefs were not arbitrary. They mirrored the biological reassurance provided by stars. The sense that order prevailed above reinforced the capacity to yield below. Sleep occurred not in isolation but within a cosmos that felt charted, structured, and trustworthy. Modern cognitive neuroscience has begun to uncover how this sense of orientation interacts with sleep. Functional MRI studies reveal that hippocampal activity decreases in the absence of environmental anchors, even when subjects are placed in virtual environments. When external cues are reintroduced, hippocampal coherence improves, and participants report greater subjective ease. Though these experiments rarely address starlight directly, the principle holds. Orientation stabilizes the brain, and the night sky was humanity's most enduring orientation system. To lose it is to unsettle a neural expectation that sleep depends upon. The legacy persists even in artificial contexts. Astronauts on the International Space Station, deprived of a natural horizon and exposed to 16 sunrises each day, often experience severe circadian disruption. Yet when they look out into space and glimpse familiar constellations, reports describe a momentary grounding effect, a return to orientation amid disorientation. Sleep quality improves after such observations, even in conditions where environmental cycles are chaotic. The presence of stars restores a frame of reference, suggesting that the brain still relies on them as a stabilizing force despite environments vastly removed from ancestral contexts. This connection between stars and rest carries implications for psychological resilience. Military studies on soldiers deployed in desert or mountain terrains note that sleep is often deeper and less disturbed when troops can see the open sky compared to when they are enclosed in tents. Orientation reduces hypervigilance, allowing the body to release its grip on consciousness. Even when threats remain, the visible stability above communicates continuity, a message that calms the nervous system enough to permit essential recovery. The stars, in this sense, are not just markers of location but signals of continuity in an unpredictable world. The absence of this orientation produces consequences that extend beyond individual rest. Urban environments, where light pollution obscures the night sky, generate populations that grow up without habitual exposure to stars. Studies on city dwellers show higher rates of sleep disorders, anxiety, and circadian misalignment, outcomes often attributed to artificial light alone. Yet the loss of orientation may contribute in ways less easily measured. A brain deprived of external anchors learns to rely solely on artificial cues, which shift, flicker, and destabilize. The nervous system, conditioned to expect permanence above, encounters only variability, leaving rest unsettled even in darkness. The capacity to fall asleep is never guaranteed. It depends on trust, orientation, and signals that the environment is both secure and continuous. The stars once supplied this assurance each night, embedding themselves into neural pathways and cultural traditions alike. What remains unresolved is whether their absence leaves only a gap in the sky or something deeper, a lingering state of disorientation within the brain that resists quieting, even when the body seeks rest. Sleep demands a paradoxical state. The body must let go of vigilance in order to restore itself, yet the brain evolved in environments where danger was ever present. This tension is resolved through safety cues, signals that communicate to the nervous system that it is permissible to yield. The night sky, filled with stars, became one of the most consistent and enduring of these signals. Their stability communicated order, their repetition across seasons implied continuity, and their silence suggested the absence of immediate threat. The neurobiology of safety is mediated primarily through the interplay of the amygdala, hippocampus, and prefrontal cortex. The amygdala scans for danger, primed to trigger fight-or-flight responses even during light stages of sleep. When cues of safety are present, prefrontal regulation dampens amygdala hyperactivity, reducing the likelihood of nocturnal arousals. In ancestral environments, stars functioned as a reliable external reassurance. They were not subject to sudden disappearance, unlike the flickering of firelight or the unpredictable movement of predators. This constancy provided a framework against which the amygdala could stand down, lowering cortisol and facilitating transitions into deeper stages of rest. Oxytocin, often associated with bonding, also plays a role in modulating safety perception. Studies reveal that oxytocin levels rise in environments perceived as secure, enhancing parasympathetic activation and reducing cardiovascular stress. The collective experience of sleeping under the stars, families, tribes, and groups gathered in common areas, may have amplified oxytocin release, weaving physiological calm into social connection. The stars were the backdrop against which communal rest unfolded, magnifying their role not only as navigational markers but as neurochemical facilitators of safety. The locus coeruleus, a brainstem nucleus releasing norepinephrine, further illustrates the balance between vigilance and rest. In high-alert conditions, its activity surges, maintaining arousal and preventing the descent into restorative sleep. Yet in secure environments, its firing diminishes, allowing slow-wave oscillations to dominate cortical activity. Night after night, under a stable sky, the locus coeruleus would learn to associate starlight with reduced necessity for vigilance. This conditioned association etched itself into the biology of sleep regulation, an imprint still detectable in modern humans who find greater ease resting in open, natural settings compared to sealed, artificial rooms. The influence of safety cues extends beyond the brain to the autonomic nervous system. The vagus nerve, central to parasympathetic regulation, responds strongly to environments of perceived calm. Measurements of heart rate variability show significant increases in individuals exposed to natural landscapes at night compared to those placed in urban or artificially lit surroundings. Elevated vagal tone is a marker of resilience, allowing the body to shift efficiently between states of arousal and rest. Starlight, as a subtle yet pervasive environmental cue, may have acted as a nightly reinforcement of vagal stability, ensuring that rest was both attainable and restorative. Anthropological records highlight the universality of this association. Among indigenous groups of North America, rituals before sleep often involved directing attention to specific constellations, invoking their permanence as a source of protection. In Australian Aboriginal traditions, the dreaming is mapped onto the night sky, linking stars with ancestral guardianship. These practices reflect an implicit recognition. Orientation to the stars provided not only practical guidance but psychological assurance. To fall asleep beneath their presence was to enter rest under watchful order, not under blind chaos. The absence of such cues in modern life destabilizes this delicate neurochemical balance. Enclosed rooms, especially when stripped of natural sound and light, can heighten amygdala sensitivity. Research on urban populations demonstrates elevated nocturnal cortisol compared to rural counterparts, even when controlling for noise and socio-economic stress. One explanation lies in the deprivation of external safety signals. The brain, evolved to expect environmental continuity overhead, is left in an ambiguous state, with vigilance only partially quelled. Insomnia and restless sleep follow, not solely because of artificial light, but because the nervous system lacks confirmation that surrender is safe. This deprivation has consequences for immune regulation. The cytokine balance that supports nocturnal repair is sensitive to cortisol rhythms. When hypervigilance persists into the night, pro-inflammatory cytokines increase, reducing sleep depth and impairing tissue restoration. Over time, this contributes to systemic inflammation, a condition linked to cardiovascular disease, metabolic disorders, and mood dysregulation. What might seem like a mere absence of stars in the modern night sky may in fact represent the removal of a regulatory input that once maintained the delicate equilibrium between vigilance and rest. Neurochemical pathways also suggest why starlight is linked to dream phenomena. During REM sleep, the amygdala remains highly active, but its output is shaped by hippocampal and cortical processes. If stars provided a nightly anchor for spatial and emotional orientation, their absence may alter dream content, leaving dreams more chaotic, fragmented, or dominated by threat perception. Some ethnographic studies record that dream narratives in societies sleeping under open skies are more structured, often reflecting journeys and landscapes, while urban populations report dreams more often infused with confusion and anxiety. The environment may impress itself onto the architecture of dreams through the neurochemistry of safety and orientation. Experiments in sensory substitution further highlight the principle. When participants are exposed to environments with stable background cues, whether auditory, visual, or tactile, their sleep architecture stabilizes, with increased slow-wave activity and reduced nocturnal awakenings. The stars were humanity's most consistent background cue, a nightly sensory field so constant that the brain could calibrate itself against it. The removal of this field is not neutral. It introduces instability into a system conditioned to expect permanence. The nervous system retains this memory even when denied its source. Many individuals report a peculiar unease when sleeping in windowless rooms, despite objective safety. The unease is not purely psychological. Measurements show elevated sympathetic tone in such conditions. The absence of external confirmation leaves the body caught between readiness and rest, unable to release its grip fully. The stars, once an omnipresent assurance, are missing, and the body resists descent into the depths of unconsciousness. The question pressing on neuroscience today is whether artificial substitutes can replicate these ancient safety cues. Virtual skylights, projected constellations, and simulated night environments attempt to restore what has been obscured by light pollution and architecture. Yet the evidence remains inconclusive. The nervous system seems to recognize the difference between authenticity and simulation, responding more robustly to genuine starlight than to its replicas. This raises the possibility that the quality of sleep depends not only on the presence of cues, but on their authenticity, a condition that modern interventions struggle to reproduce. What remains unresolved is the long-term consequence of a species drifting ever farther from its ancestral sky. If the neurochemistry of safety was calibrated by stars, then its absence leaves a gap that no artificial environment has yet filled. Each night spent without them presses the question of how much deprivation the nervous system can endure before its architecture reshapes itself into something unrecognizable. The human nervous system is not shaped by light alone. It is equally tuned to the cadence of darkness. Stars provided a textured darkness, one neither absolute nor chaotic, punctuated by points of order scattered across the void. The interplay between faint light and vast obscurity engaged sensory systems beyond the visual, reinforcing rhythms that guided both physiology and perception. Neurophysiological studies on sensory integration show that the absence of one stimulus heightens the activity of others. In darkness, auditory and somatosensory pathways become more sensitive, sharpening awareness of subtle sounds and tactile cues. Yet when the sky remained visible, the nervous system did not descend into the kind of anxiety produced by complete obscurity. The stars prevented the brain from entering full sensory deprivation, offering a scaffold of continuity that moderated this heightened sensitivity. Sleep under such conditions was not a plunge into void, but a descent into a patterned environment, one that reassured the senses without overwhelming them. The suprachiasmatic nucleus, while responsive to light, also coordinates with oscillatory activity in other neural circuits. Electroencephalographic studies reveal that brain waves entrain not only to cycles of day and night, but to subtler rhythms of exposure and absence, of faint light against deep darkness. When the visual field contains constellations, the brain perceives intervals of repetition, a structured randomness that aligns with its intrinsic oscillatory patterns. This alignment enhances synchronization between cortical networks, promoting the transitions necessary for restorative sleep stages. Across cultures, rhythmic practices developed that mirrored these patterns. Chants, drumbeats, and ritual movements often took place under open skies, their timing reflecting the cadence of celestial cycles. Neuroscientists studying rhythmic entrainment find that such activities synchronize neural firing across widespread cortical regions, reducing variability in oscillatory timing. This synchronization, when followed by sleep beneath visible stars, may have deepened the brain's propensity for coherence, weaving cultural rhythm into biological rhythm. Sleep then unfolded not as isolation, but as continuity between ritualized waking activity and the ordered presence of the sky. The thalamus, acting as the sensory gatekeeper, plays a central role in this process. During sleep, it filters incoming information, allowing certain cues to penetrate while suppressing others. Studies demonstrate that predictable rhythmic stimuli, such as repetitive tones or pulses of light, are more easily suppressed than irregular, threatening signals. The stars provided a form of natural predictability. Their slow, imperceptible movement across the sky offered no irregular intrusion, permitting the thalamus to maintain its gating without constant reactivation. In contrast, environments without such order, where darkness is absolute or where artificial intrusions break unpredictably, force the thalamus into vigilance, fragmenting sleep architecture. Modern experiments with sensory isolation tanks provide a useful parallel. Individuals deprived of external cues often report anxiety, hallucinations, and difficulty maintaining a stable sense of self. The absence of patterned input destabilizes cortical networks, leading to disorientation and heightened stress responses. Yet exposure to even faint, predictable cues restores equilibrium. The night sky, with its patterned darkness, functioned as a natural antidote to the instability of sensory void. The stars prevented the nervous system from dissolving into unchecked arousal or hallucination, providing just enough structure to sustain coherence through the vulnerable state of sleep. The vestibular system, responsible for balance and orientation, also interacts with these cues. Research on nocturnal navigation in both humans and animals reveals that the vestibular system integrates visual information from stars to calibrate movement and direction. This integration continues into rest, where vestibular input influences dream content and the sensation of body position. Without external anchors, the vestibular system drifts, leading to sensations of falling or disorientation in sleep. Many cultures describe dreams of flight or vertigo, often associated with sleeping indoors without visible skies. In contrast, reports from nomadic groups describe dreams more grounded in spatial journeys, reflecting an anchored vestibular system calibrated by nightly exposure to the stars. The auditory dimension deepens this relationship. In the absence of artificial noise, natural soundscapes, wind, insects, distant water, interacted with the presence of the stars to create an integrated environment of rhythm and continuity. Research in psychoacoustics indicates that exposure to stable, low-level natural sound increases alpha-wave activity, a state linked to relaxation and transition into sleep. When combined with the visual constancy of stars, these sounds formed a multimodal safety net. Each sense contributed to the perception of stability, reinforcing the body's decision to yield to unconsciousness. Even olfactory cues play a role. Studies show that the amygdala responds directly to certain smells, modulating sleep depth. Natural nighttime environments, rich with plant volatiles and the cool air of open spaces, produced olfactory profiles distinct from enclosed interiors. The presence of stars, inseparable from such outdoor conditions, came bundled with these sensory signatures. To the brain, the scent of night air was not separable from the view of the constellations above, and both together signaled that the environment remained continuous, ordered, and safe. The modern deprivation of stars removes not only a visual anchor, but an entire sensory constellation. Artificial lighting alters circadian signals, but enclosed spaces also strip away soundscapes, airflows, and orientation cues that once worked in concert with celestial patterns. The result is a nervous system left in partial isolation, prone to restless vigilance, deprived of the coherence it once received from a patterned darkness. Each sense, denied its natural reinforcement, begins to strain toward stability, and the strain itself disrupts rest. What remains striking is how quickly these systems recalibrate when re-exposed. Field studies in remote environments demonstrate measurable changes within a single night: lowered sympathetic tone, increased slow-wave sleep, and subjective reports of calm. The nervous system responds as though recognizing a long-lost condition, one written deeply into its architecture. Yet this recognition raises the unresolved question of cost. What accumulates across decades when the patterned darkness of the stars is withheld, and what hidden thresholds might already be eroding under the strain of deprivation? The nocturnal environment shaped not only melatonin, but an intricate cascade of hormonal regulation. The interplay between cortisol, growth hormone, prolactin, and serotonin was orchestrated under the canopy of stars, each influenced by the sensory and psychological context of the night. To understand why humans once slept more deeply beneath starlight, one must trace how the endocrine system evolved in synchrony with a patterned sky. Cortisol, the body's principal stress hormone, follows a circadian rhythm sharply defined by light exposure. In natural conditions, cortisol declines after dusk, reaching its nadir during early sleep before rising again near dawn. The presence of stars provided a consistent, low-intensity environmental cue that reinforced this decline. Studies of populations sleeping outdoors without artificial lighting show markedly lower evening cortisol compared to urban counterparts. This reduction permits the onset of slow-wave sleep, during which memory consolidation and tissue repair occur. Elevated evening cortisol, by contrast, fragments sleep, leaving the body in a state of semi-vigilance. The absence of stars does not directly elevate cortisol, but the deprivation of natural reassurance allows stress pathways to remain active, preventing their nightly descent. Growth hormone, critical for cellular repair and metabolic regulation, is released in pulses during deep sleep. Its secretion depends on both melatonin rhythms and the suppression of cortisol. Under ancestral skies, these conditions aligned. Melatonin rose, cortisol declined, and growth hormone pulses reached optimal amplitude. Modern endocrinology shows that sleep deprivation or circadian disruption reduces growth hormone output, impairing recovery from injury and accelerating metabolic dysfunction. Athletes training in natural environments often report faster recovery, a phenomenon partly explained by the alignment of hormonal cascades under authentic night conditions. The patterned presence of stars may have acted as a silent regulator, ensuring that growth hormone secretion followed its evolutionary rhythm. Prolactin, often overlooked outside its role in lactation, exerts widespread influence on immune regulation and energy metabolism. Its nocturnal rise is one of the most consistent endocrine features of sleep. Research indicates that prolactin release is potentiated by darkness, but also modulated by psychological states of safety and continuity. In animal studies, exposure to unpredictable nocturnal light suppresses prolactin, disrupting reproductive cycles and weakening immune function. Human parallels are evident. Individuals in environments saturated with artificial light display attenuated prolactin rhythms, correlating with higher rates of autoimmune disorders. Under starlit conditions, by contrast, prolactin secretion was reinforced by the perception of stable continuity overhead. The hormone became not only a regulator of physiology, but an index of environmental trust. Serotonin, precursor to melatonin, also exhibits nocturnal dynamics. Daytime serotonin synthesis is enhanced by exposure to natural light, yet its nocturnal availability is influenced by the transition into darkness. Ethological studies of primates demonstrate that serotonin turnover peaks during twilight, preparing the brain for melatonin conversion as night deepens. Stars provided an unbroken continuum during this transition, preventing abrupt shifts between light and dark that might destabilize serotonin availability. In humans, the absence of stars may fracture this process, leaving serotonin metabolism more susceptible to erratic swings. Such disruptions manifest in mood disorders, where patients often report difficulty sleeping in enclosed, artificially lit environments. The interplay between these hormones reveals a broader principle. Endocrine regulation evolved not in isolation, but in dialogue with the sensory environment. Cortisol's descent, growth hormones' pulses, prolactin's rise, serotonin's handoff to melatonin, all unfolded against a background of faint, stable illumination. To remove the stars is to remove part of the dialogue, leaving hormonal rhythms to proceed without the environmental confirmations they once expected. This dissonance does not halt the processes, but it renders them less coherent, more fragile, and more easily disrupted by stressors. Archaeological evidence indirectly supports this alignment. Skeletal remains from pre-industrial populations show lower incidences of stress-related bone lesions compared to urbanizing groups of the same era. While multiple factors are at play, endocrinological stability under natural conditions likely contributed. Growth hormone and prolactin supported tissue resilience, while lower evening cortisol reduced chronic wear on bone and muscle. The stars, though silent, were part of this biological scaffolding, ensuring that hormonal cascades operated within their expected context. Contemporary field studies provide striking contrasts. When participants spend even short periods camping in light-free natural environments, their hormonal rhythms recalibrate within days. Cortisol slopes steepen, melatonin onset advances, and prolactin secretion increases. Sleep becomes longer and more consolidated. These shifts occur without pharmacological intervention, suggesting that the body retains a latent template for nocturnal regulation that re-emerges when placed under ancestral conditions. The nervous system, it seems, has not forgotten its alignment with the stars. Yet the absence of these cues does more than disrupt nightly cycles. It alters the long-term balance of endocrine function. Chronic disruption of growth hormone and prolactin undermines tissue repair, contributing to accelerated aging. Flattened cortisol rhythms correlate with increased mortality, while serotonin-melatonin imbalances predispose to depression and anxiety. Each of these outcomes can be traced to the erosion of environmental synchrony, the loss of subtle but powerful signals that once structured the night. What remains unclear is whether artificial interventions can restore this lost synchrony. Melatonin supplements adjust onset, but not the broader cascade. Bright light therapies reset circadian phase, but do not replicate the nuanced cues of patterned darkness. Hormone replacement therapies target isolated pathways rather than the integrated whole. The night sky, in contrast, provided a holistic stimulus that coordinated multiple systems simultaneously. Its absence leaves not a single void but a network of gaps across the endocrine landscape, each compounding the others in unpredictable ways. The unresolved question is how deeply this deprivation shapes human biology across generations. If the endocrine system evolved under starlit conditions, then its gradual uncoupling from those cues may not merely disrupt sleep, but reconfigure the very architecture of human physiology. The long-term trajectory of this shift remains uncertain, and with each passing year of life beneath obscured skies, the experiment continues without clear limits. The presence of stars did more than calm the nervous system or regulate hormones. It provided a cognitive framework that shaped how memory and imagination unfolded during the night. Human sleep is not passive suspension, but an active reorganization of experience, a process heavily dependent on cues that signal order and orientation. The stars supplied a nightly structure against which memory traces could be arranged, and their absence fractures this architecture in ways that extend into both dreams and waking cognition. The hippocampus is central to this process, replaying neural sequences from waking life during slow-wave sleep. These replays, compressed in time, stabilize fragile memory traces and integrate them into long-term networks. Studies of place cell activity in animals show that spatial maps are strengthened by exposure to fixed environmental cues, such as landmarks or celestial markers. For early humans, constellations provided a nightly lattice, stabilizing hippocampal replays with external references that did not shift or vanish unpredictably. The stars anchored the mind's nocturnal rehearsals, ensuring that memories were not only consolidated but situated within a coherent spatial and temporal frame. Dreams, emerging predominantly during REM sleep, draw on these same networks. The amygdala and hippocampus remain highly active, generating narratives that blend memory fragments with emotional salience. When the night sky was visible, dream content was continuously informed by celestial orientation. Anthropological studies reveal recurring patterns in the dreamscapes of cultures that sleep outdoors: journeys guided by stars, encounters framed by constellations, ancestral figures appearing in alignment with celestial positions. These patterns suggest that the stars did not merely influence navigation in waking life, but permeated the architecture of dreaming itself, embedding the heavens into the symbolic layer of human cognition. The interaction between dreams and memory is not incidental. Research on memory reconsolidation demonstrates that dreams serve as testing grounds, reweaving fragments into coherent narratives that can be applied to future behavior. Under a star-filled sky, dreams may have been more structured, drawing on a reliable backdrop that reduced chaos in narrative formation. In modern urban settings, deprived of these anchors, dream reports often contain themes of disorientation, fragmentation, and instability. The absence of celestial order leaves the dreaming brain without the scaffolding it once relied on, producing content that mirrors the instability of the sensory environment. The prefrontal cortex, largely quiescent during REM sleep, occasionally reasserts itself in lucid dreaming. Ancient traditions describe practices of deliberately using stars as points of focus to enter altered dream states. Yogic and shamanic systems, from Tibet to Mesoamerica, taught initiates to fix their attention on a star before sleep, carrying the image inward as a guidepost. Neuroscientific studies on lucid dreaming confirm that pre-sleep focus on visual cues increases the likelihood of metacognitive awareness within dreams. The stars, in this context, were more than environmental reassurance. They were tools for consciously navigating the dream realm, blurring the boundary between external orientation and internal exploration. The cultural encoding of this relationship is visible in mythologies across civilizations. The Greeks imagined heroes and creatures immortalized in constellations, the Egyptians tied star cycles to journeys through the afterlife, and the Polynesians wove navigational dream training into their star lore. These mythologies are not arbitrary storytelling, but cognitive records of how the stars structured both memory and imagination. By embedding celestial references into cultural narratives, societies preserved the role of the stars in organizing not only space but also the architecture of thought itself. Neurochemical pathways reinforce this connection. Acetylcholine, dominant during REM sleep, facilitates plasticity in hippocampal circuits. Its activity is modulated by environmental context. Enriched environments during waking hours increase acetylcholine turnover and enhance dream vividness. Sleeping under stars, surrounded by stable but intricate sensory input, may have amplified this effect, making dreams more vivid and memory integration more robust. Modern environments, stripped of natural cues and saturated with artificial light, diminish this turnover, leading to flatter dreams and weaker memory reconsolidation. Laboratory experiments lend further evidence. Subjects exposed to simulated star fields before sleep display increased dream recall and greater narrative coherence in dream reports compared to those exposed to uniform darkness or artificial patterns. The visual system, even when only faintly stimulated, appears to imprint orientation cues that persist into REM activity. These findings suggest that the presence of stars does not merely affect circadian timing, but directly shapes the structure and content of dream cognition. The implications extend beyond individual experience. Collective memory, preserved through oral traditions, may have been reinforced by nightly exposure to the same celestial archive. Groups who oriented their myths, calendars, and histories around constellations carried forward not just stories but frameworks of memory itself. Night after night, the stars rehearsed continuity for entire societies, embedding coherence across generations. The obscuring of the night sky today interrupts this transgenerational rehearsal, leaving memory to rely solely on artificial symbols that lack the permanence of the celestial canopy. The absence of stars, then, removes more than a backdrop. It erases a reference system for memory and imagination. Dreams become unanchored, memory consolidation loses external scaffolding, and cultural narratives drift from the framework that once bound them to cosmic order. What remains unanswered is how deeply this deprivation reshapes cognition itself. If the stars served as nightly architects of memory and imagination, their disappearance may leave not just an empty sky but a void within the structures of mind that once depended on them. To understand the cost of a sky erased, it is necessary to examine what happens when the nervous system confronts absence where it once expected continuity. The brain is not neutral in the face of missing information. It fills gaps with vigilance, substituting uncertainty for order. This mechanism, adaptive in environments of threat, becomes maladaptive when applied to the silent deprivation of the modern night. Electrophysiological recordings show that when environmental predictability declines, the brain's default mode network exhibits increased activity during early sleep stages. This network, typically involved in self-referential thought and mind-wandering, intrudes upon processes that should be devoted to recovery. The absence of external orientation cues leaves the mind circling inward, producing rumination, fragmented dreams, and difficulty sustaining restorative stages. Under ancestral skies, the default mode network was quieter, subdued by the presence of external constancy. With the stars obscured, self-referential loops expand, crowding the space once occupied by cosmic order. Insomnia often emerges at this juncture. Clinical studies reveal that insomniac patients display hyperconnectivity between the amygdala and cortical regions during attempts at sleep. This hyperconnectivity reflects an inability to disengage from vigilance. While artificial light and stress contribute, the absence of safety cues deepens the condition. Environments sealed from the sky deny the amygdala external confirmation that the world remains stable, leaving internal circuits to cycle endlessly in search of reassurance that never arrives. The body responds with physiological compromise. Elevated sympathetic tone during sleep raises nocturnal heart rate, increases blood pressure variability, and suppresses parasympathetic recovery. Longitudinal research links these disruptions to increased cardiovascular risk, independent of other factors. The missing stars are not experienced as an acute shock, but as a chronic absence, a nightly erosion that compounds across years. Each night of deprivation becomes a microdose of uncertainty, a stressor hidden in the very structure of the environment. Neuroimmunology adds another dimension. Natural darkness punctuated by starlight synchronized immune rhythms, allowing for efficient nocturnal repair. Cytokine expression followed predictable slopes aligned with melatonin, cortisol, and prolactin. In artificial environments stripped of natural cues, these rhythms lose coherence, with pro-inflammatory markers rising during inappropriate phases. Such misalignment weakens resistance to infection, slows wound healing, and accelerates degenerative processes. The immune system, like the nervous system, expects patterned absence, not total void. This deprivation also alters metabolic pathways. Slow-wave sleep, when coupled with stable circadian signals, promotes insulin sensitivity and lipid regulation. Modern sleep, fragmented by vigilance and deprived of environmental assurance, leaves glucose metabolism impaired. Epidemiological data confirm higher rates of metabolic syndrome among urban populations living under severe light pollution, even after adjusting for diet and activity. The stars, or their absence, imprint themselves upon the body's handling of energy, linking the night sky to the most fundamental transactions of life. Psychiatric outcomes reflect the same deprivation. Depression, anxiety, and post-traumatic disorders all exhibit heightened nocturnal disturbances. Patients consistently report difficulty sleeping in enclosed or artificially lit environments, even when objective safety is assured. The nervous system responds not to rational knowledge but to evolutionary expectation, a patterned sky. The absence of that expectation is encoded as a form of isolation, a sensory void interpreted as danger. Modern treatments target neurotransmitters with drugs, yet the absence of external reassurance remains unaddressed, leaving recovery partial at best. Historical accounts illustrate how quickly absence reshapes perception. Sailors on overcast nights without stars reported profound unease, even when ships were secure and provisions ample. Their journals describe sensations of disorientation, dread, and insomnia. In polar regions during long dark winters, communities reported similar disturbances, with elevated depression and sleep disruption correlated with periods when stars were invisible behind clouded skies. These accounts demonstrate that the absence of celestial order destabilizes both body and mind, regardless of material security. Modern architecture compounds this absence. Windowless bedrooms, high-rise apartments cut off from natural horizons, and urban light domes create conditions of deprivation so normalized that their effects are rarely acknowledged. Yet experimental reintroduction of natural cues, through skylights, rooftop sleeping, or retreats into dark sky reserves, produces immediate shifts: lowered nocturnal cortisol, increased dream vividness, deeper slow-wave sleep. These results point to a system not broken but awaiting its missing context. The deeper question remains unresolved. What adaptations emerge when absence persists across generations? Children raised without exposure to stars may never form the neural associations that once calibrated safety, orientation, and memory. Their nervous systems adapt to void as normal, but adaptation may carry hidden costs: altered sleep architecture, blunted dream cognition, heightened baseline vigilance. These shifts, subtle at first, could accumulate into traits that define entire populations, marking a departure from the physiology that evolved under the nightly presence of celestial order. The body remembers what the sky once offered, yet the memory is increasingly forced to operate without reinforcement. Each night of absence presses the question of how much can be forgotten, and whether the nervous system, deprived of its ancestral archive, begins to construct substitutes of its own. What form these substitutes might take, and how they reshape the balance between vigilance and rest, remains an open uncertainty pressing against the future of human sleep.